Welcome back, everyone. Welcome back to another episode of World at the Moment. Just before the holidays, I want to share with you some good news. Actually, the FDA has approved count them, two new gene therapies. These therapies are called Casgevy and Lyfgenia, and these are treatments that are going to use gene editing techniques to help combat sickle cell disease in patients who are 12 years and older. This is amazing news. Now, first, let's talk about what exactly is sickle cell disease. It's a genetic condition, so it's not your fault, but basically, our bodies, for a certain gene, there's a little mistake in here that affects our red blood cells. Because the thing is, in a healthy person, we all have red blood cells, but in a healthy person, the shape of a red blood cell is a biconcave disc. And I know I just said two words that are just completely what does that even mean, biconcave disc? Just think of it this way. Imagine you have a little frisbee. A frisbee is the best way I could say, or something, a circle. Imagine you have a circle, and at the center you have a little smaller dip, like a plate. And then the backside, there's also a little dip. So at the center, there's a little bit of a dip inside of it. So you can think of it like, imagine two plates on top of each other, but it's one thing. That's a biconcave disc. And that's what a healthy red blood cell is supposed to look like. But the thing is, in patients with sickle cell disease, this gene mistake, they have a mistake in their gene that affects their hemoglobin. What is that? Well, how do our red blood cells carry oxygen? They need to deliver it. But how do they carry it? Like I mentioned before. Well, that's where hemoglobin comes in. It's a protein that binds these oxygen molecules to the red blood cell. And with patients in sickle cell disease, they don't have really proper hemoglobins. They're a bit defective. Because of that, you create this red blood cell shape. That's not our healthy, two plates stacked together, this kind of healthy biconcave disc. We end up getting a red blood cell that looks like, a sickle. When you're working in the fields, it starts to look like a sickle, which is why it's called sickle cell disease. Now, this becomes a problem because these sickle cells essentially are just little red strings floating in your blood vessels. So it's a lot easier for these little strings to kind of clump up together and form clots in the body and actually reduce the amount of oxygen you absorb because it's like a much smaller shape. So you're at a higher risk for getting things like a stroke or a heart attack, or just in general, you just cannot perform as well because you're not absorbing as the amount of oxygen that you need. But then comes in Casgevy and Lyfgenia. So, what exactly are these treatments going to do? Well, Casgevy is going to use CRISPR-Cas9 technology. Now, no, it's not something from the fridge. This is an enzyme, CRISPR-Cas9. It's an enzyme that was discovered by Jennifer Doudna and Emmanuel Charpentier. And if you look at it, I think in 2020, they won the Nobel Prize for their achievements. So really great stuff. But what they discovered was they found out that the bacteria themselves had developed an immune system to pathogens. Yes, bacteria can get sick. Bacteria, just like humans, do deal with viruses, and they're often called macrophages. So what these two scientists discovered is these bacteria developed an immune system that genetically modifies whatever kind of material that the virus injects. Because think about it this way. What does the virus do? It injects some sort of genetic material such that it hijacks the cell machinery to produce more copies of the virus. So what this bacteria's immune system did is it just snipped that. Just snipped the gene and that's it. Boom. So what they discovered is we could take this simple gene clipping sort of technology to help us use it to modify genes. But how exactly does this work here? So let me tell you guys, it's really, really cool. CRISPR enzymes, what you need to do is you take a little smaller RNA strand, and we often call this a single guide RNA sequence or sgRNA sequence. You can think of this like a GPS for this enzyme. Because think about it, it has to navigate through trillions of base pairs. Especially if you're talking about a human genome that we're trying to edit, is to go through trillions of base pairs and try to find that exact little target thing that we want to cut. So yeah, this single guide RNA sequence helps it guide through that genome. Because what's going to happen is that enzyme is going to try to find that target sequence. And when there's a match, when that target sequence kind of matches up or links up with that RNA sequence, then the enzyme knows that this is where they should cut. So they will cut the genome, and now you have it in 2 pieces. Okay, well now your genome's broken. So what your body's going to try to do is it's going to try to repair this. Okay, we're not trying to, we don't want broken genes here. It's going to affect the organism. So our body's going to do whatever it can to repair that sort of breakage. But that's where scientists can come in and put in the editing part, where they can introduce a sort of template sequence to help the repair cells who are working to actually fix that breakage. It can help serve as a guide. That template sequence can serve as a guide, telling these helper cells how exactly to repair that break, such that you get a healthy sequence and not a defective sequence. That's how CRISPR-Cas9 is so powerful. It's just way easier and way more cost efficient than previous techniques. So now, what Casgevy is going to do is very simple. You have a patient, and the patient is going to have in his bone marrow some stem cells. Now what these stem cells are is these are cells that can turn to whatever cell that they want, that they're needed to be. So they can turn into an eye cell, a stomach cell, or even a red blood cell. So what we're going to do is, with Casgevy, what they're going to do is they're going to extract these stem cells from the patient. They're going to extract the stem cells from the patient and then use CRISPR-Cas9 technology to modify those cells, because remember, all of these stem cells in a patient with sickle cell disease will have that faulty hemoglobin gene. But when we extract these stem cells, we're going to use CRISPR technology to go inside these cells and edit their genes such that we change it from a defective hemoglobin gene to an actually healthy version, a healthy sequence that codes for the hemoglobin gene. And therefore, once we do that, we just introduce these stem cells back into the body. I think, what do? We just insert them back into the bone marrow, and then they just reproduce. These healthy stem cells start to reproduce. Actual healthy red blood cells that can float around in the patient's body, and they start to recover. Now for Lyfgenia, they're using a similar approach. So what they're going to do, kind of the same thing, they're going to extract those stem cells. They're going to edit those genes again. But what they're doing is, instead of directly kind of inserting them back into the bone marrow, what they're going to do is they're going to use a virus. No, do not worry, this virus will not make you sick. What they will do is they'll take a sort of virus and remove the genetic material that actually harms humans. So they're going to remove that genetic material that is just hurting our bodies, killing our cells, just remove that, but sort of make that virus harmless to humans. Then insert our new stem cells. And then we take that virus and we insert it into the body. And what's the virus going to do? Well, we're not going to remove the gene, we're not going to remove the sequence that dictate how exactly the virus is supposed to produce. We're going to leave that in there, but we're just going to remove the harmful stuff. But the virus, this virus vector, I should say, kind of transporting these stem cells, this virus will have sequences that will tell it how to reproduce. And so what's going to happen is the virus will reproduce in the body, because it still knows what to do in that case. So it's going to reproduce in the body, it's going to spread around the body just like any other virus, like a flu or COVID, and just spread those stem cells everywhere so that they can differentiate and turn into healthy red blood cells for the patient. And there is effectiveness with these treatments. I mean, according to some of the evaluations done here, we see Casgevy has shown to have a 93.5% success rate with patients who actually took this treatment and were actually given sufficient follow-up. This is a 93.5% success rate among that population here. And as for Lyfgenia, coming in a close second, they're showing an 88% effectiveness rate. Now, I think this is really good news. because previously all we've been thinking about is more of how do we approach this problem chemically. Okay, well, maybe I could take a pill, insert some special chemical in there, maybe insert a different chemical into the body and interact the body system, things like that. We're talking about a different approach now. We're attacking new diseases that we couldn't figure out before because, well, they're just more permanent and more written, more internalized inside the body compared to, say, a simple virus or a simple a simple bacteria infection. Because think about it this way: if you have a sickle cell disease, that is internalized in the body. Like, that is something you are born with. It's a genetic condition. Just a simple drug, an antiviral or antibacterial, it won't do much for it. But now with CRISPR-Cas9, we can actually target those diseases that we just couldn't do before. Because heck, if we're able to figure out sickle cell disease, what about, say, finding a cure for cancer, or cystic fibrosis, or Duchenne muscular dystrophy? There are so many new possibilities here with CRISPR. I think it's just going to be really interesting to see how this new powerful gene editing technology will work. And I think the FDA approving the first set of CRISPR therapies for sickle cell disease, I think it's a great first step, and it's only the beginning. Well, thanks for watching another episode of World at the Moment here. If you're new here to this podcast, my name is Krish Kawle. I'm currently in high school, and I just created this podcast to help discuss different social, financial, or health issues. Just gonna bring awareness around them of what's happening throughout the world here. But yeah, if you like this episode, you can check out some of the other episodes that were made for this podcast on the YouTube channel. Once again, thanks for watching, and I hope you enjoyed this good news, and happy holidays.